All organisms are made up of cells. The organisms may be unicellular or multicellular. Cell is the smallest structural and functional unit of life. The cells of different organisms may be different in shapes and sizes, but structurally all of them are nearly similar. Cells having similar functions group together to form tissues. The animal and plant tissues are different in shapes and functions. In this chapter, we will study the structure of cell, types of tissues, the transport of molecules in and out of the cells, and so on in detail. Microscopy is the use of microscope or investigations by microscope. Those organisms or parts of organisms which cannot be seen with naked eyes are easily studied by using a microscope. It is necessary to know the two terms used in microscopy, that is, magnification and resolving power. Magnification is the ability of a microscope to enlarge the actual image of a specimen. It is represented by X. For example, if 100x is written with a micrograph, then it means this image is 100 times greater than the actual size of the specimen. Resolving power or resolution is the clear visibility of an image. It is the minimum distance between 2 points where these are viewed as two separate points. The human naked eye can differentiate between 2 points which are at least 0.1 millimeters apart. This is known as the resolution of the human eye. The compound light microscope uses two lenses and light rays, which is why it's called a compound light microscope. In this microscope, light rays pass through a specimen and are brought to a focus by two glass lenses, and then the resulting image is viewed by the human eye. One really important advantage of the light microscope is that scientists can actually observe living cells with this microscope. The light microscope can only be used for objects that are larger than 0.2 micrometers. The maximum magnification of this microscope is about 1,500 times. The electron microscope is a type of microscope that uses a beam of electrons and electromagnets for focusing an object, and that's why it's called an electron microscope. An electron microscope can enlarge images 100,000 times or even more than light microscopes. The most modern electron microscope can distinguish objects as small as 0.2 nanometers. There are two types of electron microscopes that are especially important in the study of microorganisms. The first type is the scanning electron microscope, or SEM. The SEM provides a three-dimensional view of the surface of an object. The surface of the specimen is coated with a thin layer of gold, and when electrons fall on the surface, secondary electrons are given off by the gold. These electrons are collected to produce a television-type picture of the specimen's surface on a screen. The SEM is mainly used to study the surface structure of cells. The second type is the transmission electron microscope, or TEM. The TEM gives a two-dimensional view of the specimen. Thin slices of the specimen are obtained, and electrons are passed through the specimen. The resulting image is projected on a fluorescent screen or photographic film. The TEM is used to study the internal ultrastructure of cells.